Slide set should be available in EduCat, should be posted. A lovely poem, appropriate for October. Thought this was a nice little poetic statement about mycorrhizal fungi. All right, so the plant communication section, like I said, I'll probably have more than one slide set on this, but a lot of what we've been talking about up to this point is trying to set the stage and give you background knowledge, and then we're going to start getting into more complex interactions, whether it be a lot of what I'll talk about have actually been field studies that have been done where it's a really kind of messy environment. Some of it will be lab-based as well, but um, really paradigm shifting research going on currently, and it's been a very interesting progression. I love this comment by Paul Stamets. The fact that we lack the language skills to communicate with nature does not impugn the concept that nature is intelligent. It speaks to the inadequacy of our skill set for communication. So unfortunately, a lot of scientists are not humble enough to maybe appreciate that statement. But do you all know who Paul Stamets is? The mushroom guru? That's how I guess how I would describe him. Very famous. What was that documentary? Fantastic Fungi, right? And I bought the book because the pictures in there are just fantastic. So fungi everywhere. So part of what we're going to talk about, of course, are mycorrhizal networks, because when you get into real systems, ecosystems where plants are present, you can't ignore this. And when I first got hired, I think I was the first plant biologist at Northern that focused on underground, what's going on with plants underground. And so I used to tease Dr. Robertus's grad students. I'd be often be on their committees, the grad committees, and I would always kind of push them to learn a little bit about what's going on underground with their ecosystem they were studying, because there's a lot going on that's influencing what they see above ground. And unfortunately, for many, probably centuries, that really hasn't entered the thought process of a lot of ecologists, especially non-plant ecologists. What's going on underground was just out of sight, out of mind, and was just not even considered. And that's a real shame. And now I feel so vindicated. So validated, I guess, validated, because now the research is really hot on that topic of what's happening underground. And oh my goodness, it's really influencing what's going on above ground. We had no idea. It's great to see that. Lots of new publications and symposia sessions at conferences. Mycorrhizal fungi, of course, are a big part of that, because so many connections underground are through the mycorrhizal fungal network. So Suzanne Simard is a plant biologist that works out in British Columbia, Canada, and has done a lot of work. She has written many papers. She wrote a book that was more kind of a autobiography slash life as a scientist. So she actually was a forester by training. And she talks in that book of how she went through that whole process. But once she became a forester, I think I got the impression from how she was writing about that a lot of what they did just didn't quite sit with her, that there was more to it, that they need to think a little bit differently about their forestry practices. And so she started branching out more and decided to go into grad school, get her PhD, and start looking a little differently at plants and ecosystems. And so her work it was kind of spearheading this whole idea that we have to think about ecosystems, not just everything's in competition with each other in terms of the plants competing for light and trying to kill each other and blah, blah, blah. We have to think a little differently and more carefully in this. So this, by the way, I will post this paper. It's a very good paper. And so I will post that as a reading. Just to refresh your memory with mycorrhizal fungi, we have this idea that you have different types. Typically in forested systems, you have the ectomycorrhizal fungi, you have the vesicular or arbuscular mycorrhizal fungi. They can have different morphologies and interactions with the root. Kind of looks like they're drawing here an ecto with that thick, all that gold. To me, that looks like they're trying to suggest that it's an ectomycorrhizal fungi thickly covering the root surface, but then you also have hyphae that are growing out. If you have a lot of hyphae, they tend to use the word mycelia. So it's kind of the same idea. It's just like a group of hyphae. Okay, what else? So you have hyphae that grow out into the soil. Some other terms to be aware of, rhizomorphs. So when you have, again, lots of hyphae growing in close proximity to each other, where it's almost like a tissue-like growth, then they start referring to it as a rhizomorph. Some fungi do that. I don't know that every single mycorrhizal fungus does that, but many, many do. Another term to be familiar with is sclerotia. This is the idea of kind of a resting, they're not spores, but they're parts of the fungus that can kind of go dormant and persist for long stretches of time, even in not so hospitable environments. So they see kind of all of this is looking at different ways that the hyphae can exist under different situations. We know that the mycorrhizal fungi, of course, are gathering water and important minerals and other things for the plant. In this paper, she gets into talking about networks. So I wanted to talk a little bit about that. So she compares random network versus scale free. And when you look at forest systems, she says the circles are called the nodes, and those represents the trees, okay? And then the lines that link the trees or the nodes represent the mycorrhizal fungal connections. So if you look at random networks, it says each tree node is linked to a relatively small number of other nodes that are randomly distributed. Okay, randomly distributed around the network. So very few connections from one node to the next. Well, does she say it here? These networks can be easily traversed because there are few steps or degrees of separation between the nodes. Random networks are more resilient to perturbations than scale-free networks. That's one thing I want you to pick up on, because that actually has a lot of impact on how you manage that forest. And so when you look at scale-free networks, the degree of links between the nodes is variable, where some nodes or hubs are highly connected. So when they say the word hubs, they're usually referring to one that is highly connected relative to average. This model is more representative of living networks such as mycorrhizal networks, which grow by accretion and have a dendritic form. So they just mean it's continually growing over the source or over the lifespan of that forest. So forest systems work more like scale-free networks. So in this, she's saying random networks are more resilient. So if you're a forester and you happen to go into a forest system and you think it's a random network, it doesn't matter which trees you chop down and get rid of. The health of the ecosystem is going to be fine. So this is some of her, well, not really her super early work, but this is some of the work that she was trying to get out there to foresters, that in reality, forest ecosystems aren't random. They're that scale free. So this idea here is actually from her research, where she looked at Douglas fir forest systems in British Columbia. And she was looking at the connections of the mycorrhizal fungi amongst all of the different trees that they were studying. The fungus, its genus name is Rhizopogon. That's an ectomycorrhizal fungi. So she was looking at two different species of Rhizopogon. Circle represent tree nodes sized according to the tree's diameter and colored with four different shades of yellow or green that increase in darkness with increasing age class. Lines represent the distance between the trees that are linked, so by mycorrhizal fungi. So you can see, so these yellow ones would be really young trees. And then the really dark green ones would be more like the hub trees, the old trees that have been there a long time. And then you have gradations in between those two extremes. So if you look, like this one over here has relatively fewer connections compared to, for example, this one, right? So this is a good example of scale free, not random. So this system, if they went in there, and of course, if you're a forester, basically you're trying to make a profit, right? You're going to chop down that tree and sell the wood. So you generally go for the bigger ones. And if you look, the bigger ones, the hub trees, have so many connections. They're very long lived. They're the oldest. So they're very self-sufficient, mature. They're very capable of all these interactions. And what her research is finding is that these hub trees are disproportionately more important to the ecosystem. And yet those are the trees that foresters go to first grab and get, chopped down. And so this idea is, she's saying, and she's not totally against forestry. I don't want to put it that way. That's not really the point. She's just saying we need to think about it a little differently to manage forests. And, like we still need wood and, that's not up for dispute. The issue is maybe we should think more carefully about how we manage these forests. So a lot of her work is applicable if people can hear it. Whether or not they can hear it or not, or refuse to listen is another matter. But basically, her research that she's been doing, she's done both controlled field plots where she plants these trees and over time follows their growth and interactions. She's also gone into the messier field plot sites that are just natural ecosystems. So she's done those. She's done lab experiments as well, where she works with seedlings and looks at, the introduction of certain fungi. So she's done it all. And she's finding that when they're looking at what's going on with these networks of mycorrhizal fungi amongst all the trees that they're hooked into, you definitely see carbon flow. Sugars, carbohydrate, definitely are seeing nitrogen and water. And then they're also starting to look more deeply into what else. It kind of makes sense that there's going to be nutritional substances going around, but what about more nuanced chemicals and information that might be going on? So they're looking at defense molecules. We already know that plants can emit VOCs as a way to ramp up neighbors' defense systems. We've seen that. Whether that's on purpose or not is another debate altogether. But could that be happening underground as well? Could there be defense molecules triggering nearby trees to gear up? Could there be kin recognition chemicals that are getting passed so that the plants know which are kin versus non-kin? So she's been doing a lot of work with this, and then other scientists started to get into the game on this, and the list gets longer now. So they're looking at phosphorus is a big one. There's a lot of concern over phosphorus. If you've read a little bit about that, they're worried that we're going to run out of phosphorus eventually. Maybe not in my lifetime, but in the not too distant future for growth of plants. Allelochemicals. So again, this is the idea of competition, where one plant might put out a chemical to get rid of competitors so they can have better access to resources, including light. They've looked at genetic material, including fungal nuclei. They've been looking at whether or not that is getting passed around. Hormones, sound. So auxins and jasmonic acid are hormones. We've talked a little bit about what about sound? Now they're starting to look at that a little bit because we know sound can travel through soil and rock, and certainly it travels really well in water, which is also in the soil. And then they are, looking at how does this influence the properties of what's going on underground in that forest system. How are roots behaving and foraging for these different substances that are getting passed around? How are they growing? So they're looking, lots of research going on, and they're realizing that with the help, especially of the mycorrhizal fungi, it's like that holobiont idea, because the mycorrhizal fungi are partnering up so tightly with the plants that they're able to kind of help that system, the ecosystem, and adjust to challenges that might get thrown at them, and that the microbes, the mycorrhizal fungi, are more, they can adapt on the fly a lot faster than the plant host. And so they're now realizing, hey, the mycorrhizal fungi could be super important for the health of the overall ecosystem that we see above ground. So I just wanted to get you thinking a little bit about once the mycorrhizal fungi have the chemicals, whether it's carbon or nitrogen or whatever, they're interacting with the root. So that's what this diagram is showing. They're showing a root and cross section. And I just wanted to get you thinking, so some of like the ectomycorrhizal are covering the root surface and then sending whatever chemicals and minerals and whatever they're taking up that they want to share with the plant. It's going to be going into the plant tissue. Some of it will end up into the xylem and phloem, where it can get transmitted through the plant body, through their vascular tissue. So there's certainly that internal highway with the vascular tissue, but they're also able to quickly share in the root tissue, for root growth and all of that. You have the internal tunnel systems, so to speak, that go from, in this case, root cell to root cell. And so you can imagine the fungi can quickly, because of their intimate connection with the root tissue, and then they get the nutrients or whatever they're sharing to the root, and then the roots can share that quite readily to nearby cells with the help of the plasmodesmata. Okay, so those are the internal tunnel systems that link neighboring cells together. And so this intimate growth between the root and the mycorrhizal fungus, and then the plant has ready access to these nutrients or chemicals. So they can respond pretty quickly from the use, the uptake of those chemicals by the fungus. So they want to look at what's going on in plant-plant interactions. Unfortunately, some of the earlier research with plant-plant interactions, they didn't really consider the mycorrhizal fungi at all. Like they weren't even part of the research design. So a lot of the plant-plant communication initial studies were done in like pots of plants in a lab where the soil didn't have mycorrhizal fungi. And so that's kind of, well, it makes you wonder what the research results would have looked like if the mycorrhizal fungi had been there. But so long as we understand that, but more careful research where they're thinking about the whole holobiont, the plant and its associated microbes, including the mycorrhizal fungi. They're looking at, okay, are they involved in how they're interacting with other plants? So the plant-plant communication and other organisms, are they involved in any of some of the other ideas we've already covered, like helping plants remember or learn, have agency? So she talks about agency, that plants can have agency with the help of the mycorrhizal fungi. And that basically means they can act independently and make choices. And that's what that phrase means. And her idea here is that with the help of the mycorrhizal fungi, you can see all of these behaviors, including what she describes as intelligence, and that you can't ignore the mycorrhizal fungi in these plant behaviors. So in this paper that I want you to read, she's getting into, let's think more carefully about that. So when you look at some of the earlier research, those mycorrhizal fungi often weren't even part, like I said, they weren't part of the experimental design. And so from her perspective, there is a chunk missing that we need to think about. And so much of her research has really tried to focus on that. Oh, by the way, I'll be posting at least one video of a TED Talk she gives on forested ecosystems that is reinforcing what we're talking about in here. A couple of other things I wanted to mention that are showing up in that article that are a little, I guess, maybe you've not heard of. So she gets into network theory and network neuroscience terms. So she'll use terms like crystallized intelligence, which you can think of as memory, if you want to, that's a good analogy. So when they talk about crystallized intelligence, it's similar to memory. And then they talk about fluid intelligence, which is similar to learning. So those terms you're going to see. Then she gets into the mycorrhizal networks. And she talks about, okay, we have some that are relatively weak linkages, but they develop really rapidly. They're very active, and they can respond really rapid to changing environmental habitats or situations. And so she'll refer to that as fluid intelligence, that the mycorrhizal networks, if they behave in that way, they're exhibiting fluid intelligence, which is the idea of learning. Okay. Then she'll talk about stronger links where you have, it's almost like neural networks. Like if you have a lot of neural networks connecting neurons, they would consider that a strong link. Like, so then the strong links are the mycorrhizal networks where you get more hyphal growth. So the rhizomorphs are the really thick growth of fungal hyphae. And so she says those strong links that involve complex cores or rhizomorphs by long distance exploring fungi where they go long distances, that's more like crystallized intelligence. So it's kind of similar to when they talk about when you learn something, first is very ephemeral and you don't have many connections in your brain on that topic. And then as you learn it, the connections get stronger with more neuronal links. That's kind of what she's talking about here. And that's what happens in your brain as you commit something to long-term memory. It's kind of like growing rhizomorphs in your brain. Just consider the neural networks, the really nice connections, strong connections are kind of like the rhizomorphs. So I'm trying to describe this because she gets into it in the paper. And if you've not had this kind of jargon before, it can get a little, So as you're reading the paper, it'd be a good idea to have this a couple of PowerPoints open. All right, so the idea, we've already talked about the root brain hypothesis that the Darwins have proposed back in the 1880s or so. Now she's saying, okay, great, let's take that and now layer onto it the mycorrhizal network and see if it does have a role in this idea of the root brain hypothesis. And so she's trying to, I guess, get into the messier system where it's a more natural system where you have to think about the mycorrhizal fungi because that's how most plants exist in their real life. So I found it interesting because it made me think of this quote by a famous microbiologist that, we evolved in the presence of microbes. We can't get away from them. They're always with us, whether you're a plant or an animal, and that it's so nice to see scientists now saying we have to consider that. We have to consider all the microbes that hang out with the host and understand that interaction to understand the entire organism that we're looking at. So this is a big part of the work that she's going. And so I will be discussing some of her work as we get through this material. But some of the first things that started to get asked by plant biologists was if we can see plants being able to tell the difference between kin versus non-kin. And Suzanne Simard is also doing work with this too. But I think most of us are at least familiar with the kin recognition theory, where it makes sense that you kind of, what's the word? Well, I mean, this is said better than I can say it. You protect one's genetic inheritance, so you'll try to make sure that your kin are healthy and everyone's good to go so that you can pass your DNA into the next generation. Non-kin, not so much. And so they started thinking about this idea, especially because back in previous decades, so much of the focus on ecology was competition. And so this idea that you have to tell kin from non-kin so you know who to compete against. And so they've been thinking about kin recognition for a long time. That brings up a whole bunch of questions on, first of all, can plants even communicate that, hey, I'm kin, or you kin? Yep, we're kin. Can they do that? How are they doing that? Is it done on purpose or is it eavesdropping? What's going on? So there's a lot of things that had to get nailed down and thought about. And so there's a lot of jargon out there and they disagree on some of the jargon. So there's this idea of cues and signals, and they'll talk about, who's sending the signal, who's receiving the signal or cue. What are the fitness costs and benefits to doing that? Because if you're going to send a cue, like a volatile chemical, that was an investment on your part, because you had to make that chemical and then you had to get it out of your body if you're a plant to the whoever's around. And so if you're going to do that, there needs to be a benefit to that. So they're looking at all of these ideas. Surprisingly enough, they use a lot of social, like human social sciences to get into some of what's happening with other animals and plants and microbes. They even do all this with microbes too. They get into this whole idea of signaling. They use the word exploitation. Could that be going on? Could plants be exploiting each other? Could they be eavesdropping? Could they be manipulating? So a lot of these terminologies you hear in the social sciences when you think about the sociology of human interaction. Deceit certainly happens amongst humans, and they start thinking about this in terms of, are plants doing this? And like I said, this is big in the microbial world too. They talk about microbes with this too. Honest cues versus dishonest cues. So you're going to see some of these terms as we go through some of the readings. So cues that have some cost to the sender is, they're saying if you're going to make that signal or make that chemical and send that out, it's difficult to take that. It's a cost for you to do it. So they don't think it's a disingenuous thing or an accidental thing. Selection will favor receivers that pay attention only to these expensive and therefore reliable cues. So they're saying that maybe some cues are just eavesdropping and it's not really meant to be a true communication signal. So I just want you to know there's all this debate out there on what is a true signal or not. And it can get kind of gnarly. So natural selection will favor individuals that produce a cue only when doing so increases the chance of surviving and reproducing. That's how they're currently thinking about it. And so in some instances, production of a signal is associated with an increase in the receiver's fitness. these mutually beneficial consequences are true communication. So if you are going to define it very precisely, this is what that second bullet point there is often how I see people defining what true communication is, that the sender deliberately made this VOC or chemical, deliberately sent it off so that the sender could receive it. And it is a signal that the sender pays attention to. They both benefit from it in some way. Then they say, okay, then it's true communication. But it gets kind of murky and there's a lot of nuance in the research trying to tease this apart. Is this really true communication or just eavesdropping? So just be aware, you're going to see a little bit of messiness here with some of the jargon, I guess, and there'll be arguments about what is true communication or not. So one of the first scientists that started getting into kin recognition, it's in the notes in one of these, but his name is Richard Carban. Carban. How would you pronounce that? Karban. Would it be Carban? Carbon? Carbon? And carbon? I don't know. Anyways, Richard Carbon, he's done a lot of work. He is probably, I don't know if he's retired now or near retirement. His favorite plant seemed to be sagebrush. And so he did a lot of work out west on sagebrush field sites where it was a natural ecosystem for sagebrush growth. I finally got to see my first sagebrush in its natural growth, in its natural ecosystem. I didn't get to see that until 2018. So I finally went out west far enough where I could see sagebrush. I was all excited because I remembered all his cool studies. So one of the first studies that he did is he took 30 seeds from the same plant and grew it in one pot, 30 seeds from different plants grown in one pot. And so we're talking about kin versus non-kin here and just growing them up and seeing what would happen. So this is done in a lab-based study, no deliberate inoculation of mycorrhizal fungi. So that wasn't in these are earlier studies now. And he was able to show that if the plants were not related, they were very aggressive with their root growth. And we talked a little bit about this already with the sea rocket plants doing this. And then it was shown on that documentary I asked you to watch, that if they're not related, they're pretty aggressive in their root growth. It's kind of like every plant for itself. We're just going to try and grab all the resources we can. But those that were related still grew really well, but their root growth was more restrained, which implies that they were able to recognize kin from non-kin and behave in such a way that all the kin were benefiting from their collective behavior. Because if you get too aggressive in your root system, you'll hog and starve the other ones that you're out competing. And so you don't want to do that to your kin. All right, so it was early studies that were supporting the idea that plants seem to be paying attention to kin versus non-kin. And then we already talked a little bit about those sea rocket plants along the shore of Lake Michigan that Susan Dudley was doing research on. And she discovered the same thing much later. Her research was much later, but she did a similar study and it was the same idea, how the siblings from the mother plant in one pot, they were much more restrained in their root growth. Of course, once these studies were done and the results were implying that they can tell the difference, then they got into, okay, how are they doing this? And there were a lot of options being explored. Certainly root exudate seems to be a good one, that they're chemically communicating via that way. The VOCs, that makes sense too. They started to look at phytochrome ratios to see if they're able to sense how near their neighbor is, who their neighbor is. We'll talk more about that. They even were looking at the ability of the plants to capture blue light, which cryptochromes we talked about are the one of the big ones for blue light. They even started looking at this hormone ethylene. So ethylene is one of the plant hormones. That one in particular they were looking at because it's gaseous, so it could be a VOC in that regard. They've done previous studies where if you have a lot of plants growing together, there seems to be higher concentrations of ethylene getting released. They just picked up on that during some early studies. And so the ethylene concentration goes up when you have a dense stand of plants all growing together. And so they started thinking maybe the concentration of ethylene is somehow involved in this. They noted that if you have low red to far red phytochrome ratios, that you get an upswing in ethylene synthesis. So they started to look more carefully at that. Ethylene too, if you get a decent enough concentration of ethylene, one of the things that it does is it causes leaves to fall off the plant. So leaf senescence just means leaves are getting, so the idea here would be maybe if they're kin, one of the hypotheses was if they're kin and they're shading each other, maybe they're like self-pruning in a way through this ethylene exposure, so that they can all kind of get along with light capture. And they also know that ethylene can trigger stem elongation, right? So lots of different things were, once they came out with some of the basic research, they're like, okay, it does look like there's something going on here, but now they got to narrow it down and try to figure it out. All right, we'll stop there and continue on with this on Monday.